It is 9:01. Should we get started, Dr. Mustelin?
Go ahead.
Okay. All right. Hello, everyone. Thank you for joining the University of Washington Division of Rheumatology Grand Rounds. A couple of reminders before we start the session. This session will be recorded and will be posted later on our website. There will be a Q&A session at the end, so please add your questions in the Q&A feature and not in the chat box so that it's easier to moderate. And now I'll let Dr. Mustelin introduce our speaker for today.
Okay, thanks, Danielle. So it is a real pleasure and honor to introduce today's Grand Rounds speaker, Caroline Grönwall, from the Karolinska Institute in Stockholm, Sweden. So Dr. Grönwall got her doctoral degree in 2008 in molecular biotechnology and went to do a postdoc number of years with Greg Silverman, a very well-known B-cell immunologist from across the street in San Diego, where I worked as well. So she's spent, I think, 6 years altogether with Greg Silverman, who during that time moved to New York. So Dr. Grönwall got views of both west and east coast of the US during her stay there. So coming back to the Karolinska as an assistant professor, she was promoted to associate professor in 2019 and now runs a research group in the larger division of rheumatology led by Lars Klareskog, who we all met about 6 months ago. So Dr. Grönwall's research has focused on antibodies, antibody engineering, and understanding autoantibodies and what they look like and what they do and why they are around and their role in disease. She has published a long list of very important papers in the field, mostly focusing on cloning patient, so rheumatoid arthritis patient antibodies specific for citrullinated epitopes, as well as a few other post-translational modifications, and then studying their properties and trying to understand them in the context of rheumatoid arthritis. So she will talk to us today about autoantibodies to protein modifications in rheumatoid arthritis, disease relevance, and B-cell biology. So please go ahead, Dr. Grönwall.
For this invitation. Thank you for this opportunity to talk about our research. So I'm going to talk about autoantibodies to protein modifications in RA. And I hope that I will be able to convince you that these autoantibodies and autoreactive B cells recognizing protein modifications are very interesting and have a lot of unique features, and maybe not exactly following the immunology textbook about what we know or what we think about autoantibody or antibody recognition and B cell selection. And as you all know, seropositive rheumatoid arthritis starts many years before disease onset. We have genetic predisposition and environmental triggers. And we are discussing a lot about the mucosal interface, where we think the break of tolerance is happening years before disease onset. We are very interested in the risk phase, so where you start having autoimmunity and autoreactivity, autoreactive B cells and T cells, but not have clinical disease. But as these preclinical or risk phase progresses, the patients or the individuals start having some pain and tenosynovitis, for example, just prior to disease onset. We think a lot about what's happening in the evolution of the immune response in this phase. So That's something to keep in mind when we discuss mostly what's happening in kind of the chronic phase today. And the two major autoreactivities in rheumatoid arthritis is rheumatoid factors, so RF, anti-IgG-Fc, and the anti-citrullinated protein autoantibodies, or ACPA. And these two autoreactivities, we do think that they come from different types of B-cell responses, where the rheumatoid factor, which is less specific for RA, may come more from a T-cell independent type of simulation and have more limited germinal center interactions, while the ACPA responses have very high levels of somatic height mutations in the BCRs. We'll talk a little bit more about that, which then suggest extensive germinal center responses. These two, so the rheumatoid factor is very important, and we know that there are synergies probably between these two autoantibodies in the RA pathogenesis, where the IgM rheumatoid factor may amplify the inflammatory effect of the ACPAs, and both of these responses in B cells are interesting in the RA pathogenesis and evolution of the immune response. However, today I will focus mostly on the ACPA and the citrulline responses and the responses to other protein modifications. But the rheumatoid factor is still important, so let's not forget about it too completely. The ACPAs, then the anti-citrullinated protein autoantibodies, are recognizing citrullinaton, which is post-translational modification of the amino acid arginine that is converted into citrulline by the PAD enzymes. There have been many different citrullinated antigens or citrullinated peptides that have been identified that can be targeted by these autoantibodies. And every researcher have their favorite one. It can be vimentin or fibrinogen or alpha-enolase, collagen, and so on. And we talk about that the patients have different ACPA fine specificities, then having different autoreactivities to different citrullinated peptides originated from different antigens. And we'll talk quite a bit about that. And all of these can be captured now by the synthetic peptides, the CCP2 or CCP3 clinical kits that are designed to capture the citrulline reactivity in patients. So this is an example of what the autoantibody profiles look like in RA patients, and this is brand new data looking in 4,000 RA patients, but previous data that we published is quite similar, which is that we can see a heterogenic pattern of the autoreactivity in the patients. And in this particular study, we have used two different isotypes of CCP2, both IgG and IgA. We are including all the rheumatoid factor isotypes, both IgM, IgG, and IgA. We do see that there are some patients that have an IgG response and not an IgM response in rheumatoid factor. So including all these isotypes captures more patients. And then we are looking at the ACPA fine specificities. In this case, we have included 15 different citrullinated peptides originating from different antigens, for example, then the tenascin, fibrinogen, filaggrin, and vimentin. And you can appreciate that we have some different patient subsets here. And if we look at the seropositivities in these patients, if we include only the CCP2 IgG test, we have around 70% of patients that are seropositives. If we also include the rheumatoid factor, this seropositivities are going up to 77%. But if we also include an ACPA fine specificities, the seropositivity actually goes up to 83%. So there are some individuals that we are not capturing with the traditional tests, but they still have some autoantibodies. We have also spent some time in looking a little bit more on the details of the citrulline responses of the ACPA fine specificities. And some of the peptides that are kind of, traditionally used or that people like to use, they have multiple citrulline residues, and you know, for example, this vimentin peptide has 3 different citrulline, and this is kind of the peptide that is published very often with having all these different citrulines. But when we look into the details of the reactivity by using peptides that have only 1 citrulline residue, but you know, the original backbone, so to say, of these peptides, we can see that there very often is one of the citrulines that are dominating. So the autoreactivity is not towards the full peptide, but rather to one or the other of the citrulline or the smaller epitopes. And we can see that sometimes all of these reactivities or all the patient reactivities are captured by the original peptides, but in some cases we actually find better capture with the single citrulline peptides. And if we look at this fibrinogen peptide, you can see here that the original peptide, you know, captures quite a few of these patients. When we're looking closely, we can see that these patients here, they're really reactive to this second citrulline, while these patients here, they're really reactive to the first citrulline. If we look in the vimentin peptide here, that is another example, we can see that absolutely most of the reactivity that is seen and captured with the original peptide is coming from the first citrulline, which is really dominating the response in the patients. So we'll keep this in mind when we're looking more into some of the other data later on. So why do we have autoreactivity to citrullination? And there is maybe not a very good response to that or a very good answer. We know that citrullination is something that is naturally occurring, but increased during inflammation, and there is high level of citrullination in the RA synovium. It may also be increased by smoking. We know that there's a connection with smoking and RAs, and smoking being an environmental factor triggering RA. The same is discussed with periodontitis, where we may have citrullination by Porphyromonas gingivalis that has their own enzyme, the PPAD. And there's a lot of discussion about neutrophil activation and neutrophil extracellular traps, which can be a source of citrullinated antigens, for example, citrullinated histones that we're discussing a lot. So, but in RA, there's not only autoreactivity towards citrullation, we can also see autoreactivity towards other post-translational modifications, and we talk more and more about anti-protein, anti-modified protein autoantibodies, or AMPAs, and we're looking a lot at the autoreactivity towards carbamylation and towards acetylation, which are both lysine modifications. I'll touch a little bit on a kind of an outlier in these, which is the MDA or MAA autoreactivity, which is also a lysine modification that looks quite different. So these are all, you know, processes that are regulated differently. Some are more chemical, some are more enzymatic. They are converting a positively charged amino acid into neutrally charged, but that's maybe the most what they have in common. But if we then look at the reactivity in the patients, we can see that RA patients have quite a bit of reactivity towards carbamylated antigens. We also have a lot of reactivity towards acylated epitopes, and this is using the organ tech ELISA, which is based on the vimentin peptide backbone, where the citrulline in the peptide has been replaced either with the homocitrulline, the carbamylation, or acetylation. So this is a very good assay to directly compare the reactivity without influence of the kind of protein or peptide backbone. If we look a little bit closer on this, we can see that most of the reactivity in these patients are actually found in the CCP or ACPA positive subset of RA patients. It's maybe a little bit of reactivity in the ACPA negative RA patients, but really a majority of the reactivity is seen in the CCP positive. And we can see that there is, you know, citrulline reactivity is most frequent. there is a subset of RA patients that have also carbamylation reactivity in addition to citrulline reactivity. And there is also a patient group that has both carbamylation reactivity and acetylation reactivity. And there are not so many that have these reactivities by themselves. We can also see that there is a strong correlation with the number of citrulline reactivities in the patients, so the number of ACPA fine specificities that were positive. And we can also see a correlation with the CCP levels for these patients, with higher CCP levels, higher probability of having positivity for carbamylation acetylation. So a little bit of a sidetrack also, thinking about IgA ACPAs. So we are quite interested, or a lot of people are quite interested in IgAs. It's an interesting isotype. It's the most abundant isotype in the body because we produce very large amounts in the mucosa. So it's strongly connected to the mucosa. However, you should remember that the IgA that we detect in the blood is mostly produced by long-lived plasma cells in the bone marrow. And another thing to think about is the IgA class switching. So when looking at class switching, it's a one-way street where you go in only one direction, right? Going from IgM and to IgG or IgA, and the IgA class switching is mostly coming from IgM and IgG1, and you can't really, or you don't really go very much from IgG to IgA. There is a possibility to going from IgG2 and IgG4 into IgA2, but you can't really class switch, which also the other way around. And in chronic responses where you have a lot of B cell activation, you can maybe think that there would be a higher probability of the B cells getting pulled into in a class switch reaction. Class switching is regulated by different mechanisms, but this is something to keep in mind. The more immune response you have, also maybe the more class switching. And what we see in the IgA positivity, looking either at CCP IgA positivity or using the OrganTech modified Vimentin peptide positivities, that most of the IgA positivity in the patients that we were looking at were within IgG positivity. So there were very few that were IgA positive without being IgG positive. And we can, similarly to what we saw with the carbamylation and acetylation, we see that there is a correlation with having high IgG citrulline titers and having IgA towards the same citrullinated peptide or the CCP. So this would maybe suggest that this is a effect of chronic responses. However, we can also see that there is a correlation with smoking, and smoking, of course, when we think about mucosa, where we can see that the patients that are IgG positive and IgA positive, so double positives, compared to the ones that have only IgG positivity, they have a higher frequency of history of smoking. So there is a connection with smoking and being IgA positive, which still suggests then a mucosal interaction. So maybe it's a little bit of both when we see the IgA, both a sign of having a strong immune response or chronic response, and a mucosal interaction. So a couple of slides then on another favorite topic of mine, which is the IgA or the anti-MDA reactivity. And you all have some autoreactivity to malondialdehyde. And malondialdehyde is an oxidation-associated protein modification. It's an aldehyde that is getting released during lipid peroxidation, during excess of reactive oxygen species, which can modify the proteins in two different ways, either this linear MDA modification, or together with acetaldehyde, these circular MAA modifications. And the anti-MDA natural, meaning that at birth, we all have IgM anti-MDA autoantibodies, which are actually protective and helping us to clear modified proteins and maybe cell debris. While you're not supposed to have IgG anti-MDA, and that is elevated in inflammatory diseases and autoimmunities like lupus and RA, and correlates with disease activity. So this is not an RA-specific autoimmunity, but it is increased in RA. So this is showing the IgM anti-MDA MAA reactivity in neonatal, so at birth in cord blood, where we can see that we have a very high proportion of the IgM at birth being targeted to MDA MAA reactivity. But then we can see the IgG reactivity is lower in healthy individuals, or this is actually population controls, than it is in ACPA positive RA in particular, and also in lupus, and we can see a correlation with disease activity. We can also see that this reactivity is behaving differently from what we saw with the carbamylation reactivity and acetylation reactivity, where we in our hands don't really see an increase in risk individuals. So these are ACPA positive individuals that have not any signs of arthritis, but they are at risk of developing arthritis. And we do not see an increase in MDA MAA levels until the arthritis onset. So in ACPA positive individuals, when they are getting the first signs of arthritis, then we see an increase, and we see an increase in early RA patients. We don't see as much of a correlation with the number of ACPA fine specificity as we saw with the carbamylation acetylation reactivities. So these are really quite separate reactivities and not associated in the same way with citronellation. So to understand these different reactivities a little bit better, we needed to look into individual B cells. And I think this was really important to understand how this observation that we see in serology, how this really translates to what's happening on the B cell level. We needed to look at the individual B cells. And there's been many different efforts and large collaborative efforts to try and understand the single cell repertoires in RA patients, where different methodologies have used either flow cytometry sorting in an unbiased way, or sorting the antigen-specific B cells. We've also used single cell transcriptomics to identify different B cell subsets to look at the repertoires. And I will talk a little bit more later on about the kind of overall sequencing and the repertoire studies. But we've also done from all these different projects and different tissues and different stages of disease, is that we have cloned the heavy and light chain sequences from the B cell receptors from the B cells, and expressed recombinant antibodies and made monoclonal antibodies. And from all these monoclonal antibodies, and I wrote 500 there, I think it's more than that, we have identified CCP2 positive, ACPA positive clones. And these are really very carefully validated so that they will not have any reactivity to native antigens or to have any kind of unspecific stickiness or polyreactivity. And we now have clones that are coming both from the risk phase, from the lung of RA patients or from risk individuals. We also have a number of clones that come from exactly pretty much on the day of RA diagnosis. So very early disease, both from the lung and from the synovium. And we have a number of clones that are coming from established RA patients, coming both from the bone marrow. So we looked at the plasma cell compartment in the bone marrow, looking in the blood, in the memory B cells in the blood, and looking in synovial and plasma cells, or antibody secreting cells, and a few memory cells as well. We also have made monoclonals coming from gingiva tissue from RA patient or periodontitis patients. in this compartment, we have not found any CCP positive clones yet. We do have a number of citrulline reactive clones, and this is the only place where we have found citrulline reactivity without having CCP reactivity. In all the other cases where we have screened the antibodies for reactivity to citrullinated peptides, those that have been positive for peptide, they are also CCP positive. I will not talk so much about the MDA reactivity, but we also have identified MDA and MAA reactive clones among these recombinant antibodies. So we also have monoclonal antibodies targeting these type of modifications. So if we look at the ACPA clones that we have identified, these have several fun things going on. One of them, if we look at the subclass and the isotypes that are used, they are mostly IgG1, so no big surprise. But we also find a number of IgG4 positive clones. And IgG4 is really quite unusual in many responses, but we know that ACPAs can be IgG4. And again, I think this is a sign of chronic responses. We also have a number of clones that are originated from IgA positive cells. These all have very high levels of somatic hypermutation, indicating that they have undergone multiple germinal center rounds. And they are not exactly as high as HIV binding antibodies, but almost, and they're much higher than what we see in regular kind of vaccine responses or infection responses. Again, maybe a sign of the chronic responses. They all also, most of them have introduced N-glycosylation sites, so somatically N-glycosylation sites that are introduced by somatic hypermutations. And this is also consistent with what we know from the literature, where our Leiden colleagues first showed that there was a mass shift in polyclonal ACPA purifications, where the ACPAs have a higher mass, indicating that they have extra sugars or Fab glycosylations in the variable regions. And then that has been supported by many studies looking at the sequencing data from ACPA positive cells, both from us and from others. And also we know that the recombinant antibodies that were produced, they actually carry these Fab glycosylations as well. In our hands, we don't see very much effect of the glycosylations on the affinity. So we think that this is mostly maybe a sign of B cells, something that's happening in the B cell selection. So maybe the glycosylations are promoting B cell survival. But I think we still have some work to do to understand these glycosylations. Since we have found that all clones pretty much have high somatic hypermutations and Fab glycosylations, we have also used that as a criteria for expression of the recombinant antibodies, because expressing recombinant antibodies is a lot of work, and we want to increase our probability to find ACPA positive clones, since that was the goal in these projects. So in both when we looked in the lung B cells and the bone marrow plasma cell B cells, we selected the clones to be expressed based on having high somatic hypermutation and Fab glycosylation. So what is important from this study is, maybe we increase the probability of finding ACPA positive clones, but most importantly, you have to remember that all clones that have Fab glycosylation or high somatic hypermutations, they are not ACPA, because even though we expressed all these clones, we only identified a few that were actually CCP positive. So there are lots of clones that have glycosylations that are really part of other types of responses. So this is, ACPAs are enriched for Fab glycosylation, but it's not a unique feature of ACPA or from immune responses perspective. There's also other responses where you get it. All right, so looking at the reactivities of these antibodies, we found that all of them were multi-reactive. So they all bound to many different citrullinated peptides. Some had a more restricted binding, binding to fewer peptides, and some have a very extensive binding to very many peptides. And we cannot say that how many peptides that the monoclonal antibodies bind to is related to where they come from, or what type of B cells that they're originating from. So even if there was no trends saying that, memory B cells versus plasma cells versus double negatives would have a different type of reactivity profile. There are a couple of these clones that come from risk individuals from the lung, and they do have less multi-reactivity. They recognize they're CCP positive, but they recognize very few peptides. So, but it's a little bit hard to say if that is something that will translate into other patients and other clones as well. So we in the previous study here that I've shown, we used an array from Thermo Fisher, or that we've developed in collaboration with Thermo Fisher using citrullinated peptides from our favorite antigens. But we have also done some studies using a larger array with more than 50,000 different peptides. And then we can really see the extent of multi-reactivity that is really going far beyond our kind of favorite autoantigens. And each monoclonal antibody actually recognizes hundreds and sometimes thousands of different citrullinated peptides, and from a lot of different antigens that are not necessarily the antigens that we are thinking of in the context of RA. And we really don't know exactly which antigens that are most important in the pathogenesis, but we do know that they are recognizing many different antigens that are not our kind of standard antigens that people are looking at. And they do this by recognizing very small epitope, very small motifs that then can be present in many different antigens. We have identified these by taking the peptides that are positive on these big arrays and looking for the consensus motifs. And this is showing two different clones. So this is the positive citrulline reactivity. And you can see that there is not really any arginine reactivity to the equivalent peptides. And when we look at the consensus motifs, these are two different ones. And here are a number of different clones. You can see that there's a lot of glycines here. So that is something that we see and we know from also from other serology and polyclonal data that there is a lot of reactivities to peptides that are containing glycine in the +1 or -1 position. But there is also motifs that are recognized that do not have glycines in them. These clones, some of them also binds to carbamylated and acylated peptides, and explaining what we see in the serology of RA patients. So this is really a flavor of the citrulline reactivity. And this multi-reactivity towards carbamylation and acylation is just as extensive as what we see with the citrulline. So we do have just as many peptides that are recognized, and this is also epitope dependent. So I try to use the word multi-reactivity and not cross-reactivity, because if you think cross-reactivity, you think that you have the citrulline reactivity first, and then you have some reactivity to carbamylation or acylation, but that doesn't seem to be the case. And in particular, antibodies that recognize acylated epitopes, they really seem to have a preferential for the acylated peptides in favor of the citrulline peptides. These antibodies, they also have a strong recognition of acylated histones, which gives them an ANA binding. So they bind to nuclear citrulinated or acylated histones, which gives them a HEP2 ANA staining for these antibodies. They also bind to neutrophils and activated or nets and activated neutrophils. A lot of the AMPAs, we will get to that, binds to nets and neutrophils, but with a little bit of different patterns. So the clones that binds to acylated histones, they seem to have a little bit of different reactivity patterns compared to clones that have only citrulline reactivity and no acylation reactivity. So there are some differences in how they bind. During certain conditions, the acylation reactive anticlones will also bind in a pad independent way. So they will still bind, even if there's presumably no citrullation. These clones also bind to apoptotic cells. And if you do an immunoprecipitation, it is the acylated histones that are pulled out, not the citrullinated histones, even though there are citrullinated histones. So they are really binding with a higher affinity to the acylated peptides than citrullinated peptides, but they're still strongly CCP2 reactive. So the clones that have these multi-reactivity that extends to carbonylation acetylation, they do also seem to have a little bit of more citrulline reactivity, so broader multi-reactivity, and maybe a little bit of more somatic type mutations, making you think that this is a part of the evolution of the immune response, so that you're gaining more multi-reactivity, including carbonylation and acetylation. So then to one of the big questions, so are AMPAs pathogenic? And what we've done then is using these monoclonal antibodies that we developed for studying AMPA functionality. And as I said, many of the antibodies, but not all of them, binds to activated neutrophils and nets. They bind intracellularly, so they need to be have the cells be fixed or permeabilized, at least in flow cytometry, although you have a little bit of bias there, that you kind of, what you pull through the flow cytometry needs to kind of be intact cells and not fully cells that are fully in netosis possibly. But we do see higher binding to the fixed and permeabilized cells, and you see quite some differences between the different clones. They have also been used in a number of in vivo experiments, and if you combine two of our favorite clones, and these are with murine constant regions, so we make them chimeras for the in vivo experiment. This combination in the mice give pain or pain-like behavior. We also see that they induce bone erosion or bone loss in the mice. And we can see that they induce tenosynovitis, and tenosynovitis is something that is studied quite a bit, because there's also something that is seen in risk individuals just before onset of disease in the Karolinska cohort. So these two, combination of these two antibodies together, they do induce some phenotypes in the mice. However, if you give them alone, they do not give any arthritis. So this is giving either the monoclonal AMPA, these two monoclonals in combination that you saw in the previous experiment, or anti-collagen antibody. So this is the regular Kaya protocol, or collagen antibody-induced arthritis model. And there's really no arthritis from these AMPA clones. But if you give them together, so you give the anti-collagen antibodies together with the AMPA antibodies, you actually block arthritis. So we are not inducing arthritis, but instead blocking arthritis. And this is not seen by all clones, but some clones, and one example is this clone and CO3 that actually was one of the clones that were included in the previous experiment as well, giving pain and bone loss in combination. But here it completely blocks the arthritis in the collagen-induced arthritis or antibody-induced arthritis model. And there are some that gives a little bit of effect here, and there are many that do not have any effect. And these antibodies, they can all that have these properties, they also have a strong therapeutic effect. So even if you already have a full-blown inflammation, if you give the antibody, the AMPA antibody, the inflammation dramatically drops. So it has a therapeutic effect. There's lots of studies that are needed still to understand really the functionality of this. We can see that it's FC dependent. So if we give a Fab version, or actually a GR-LR version, which is a version that doesn't bind to FC receptors, possibly also have some complement deficiency in complement binding deficiency. This has no effect in the model, so the mice still get arthritis. What is interesting is that if we give purified polyclonal AMPA, so anti-CCP2 purified ACPA from RA patients, that preparation also have an anti-inflammatory effect, which is seen here in the blue line. And this can be compared to the flow through of the column, so IgG from RA patients that are not CCP reactive, which is not does not have this effect. So if we summarize this, we have identified a number of clones, 4 different clones in currently that have these strong anti-inflammatory effect. We do have a lot of clones that have no effects, and we have a couple of clones that actually enhance arthritis. And this is one example here. This CO5 clone that is enhancing the inflammation in the mice when given together with the anti-collagen antibodies. This has been shown by 3 independent groups. So in parallel, during 2023, there has been 3 different publications showing these anti-inflammatory properties in very similar experiments that we all started without knowing anything about it. We maybe have the most different clones, but the results are very similar. In our hands, we cannot detect or determine the binding reactivity that is responsible for these features. So we see the multi-reactivity patterns, and since we have so many different clones that have different properties, it's very difficult to see what these actually have in common that have the different features. But we have some ideas, and we're working on it. We can also not really see a connection with certain cell binding or activities in the different studies that have been that these monoclonals now have been used in. And you can see this very simple heat map here of summarizing data from many different studies. There's a lot of gray fields here, so we still have a lot of work to do to fill out the blanks here to try and understand what all these clones may do. But it is a very interesting time to work on this. So to summarize a little bit, what we've done then is we have looked at the functionality of these clones. What we can see is these very interesting features: the multi-reactivity, high level of somatic mutation, and Fab glycosylation. So this tells us that there is something interesting going on in the B cell response and the B cell biology and B cell selection. So I want to share a few slides here at the end, looking at some global shifts of the repertoires in RA patients using these single cell data and other data that we have generated. So first of all, what we can see in the RA patients is that we can also see a change in subclass and isotype frequencies, when we're looking in some big repertoire data from the bulk of RA patients, positive RA, we can detect IgG4 positive cells. IgG4 again is very rare in healthy individuals in the blood, but it's easily detected in the RA patients. We can also detect IgG4 positive plasma cells in the bone marrow of RA patients. Another interesting thing is that we can see a lower frequency of IgA positive cells, and we can see that in the blood, and we can see that in the bone marrow, and we can also see that in the lung. So not sure exactly where the IgA cells are going. We do see that we have higher levels of serum IgA, but then lower frequency of IgA positive B cells. We can also see a global shift in the N-glycosylation in the RA patients. So we can see that we have a higher frequency of N-glycosylation when we're looking at B cells in the lungs, also in the risk individuals that have not yet developed RA. We can see that in the plasma cell compartment in the bone marrow, and we can see that in the bulk repertoires in ACPA positive RA patients. And this cannot really be explained only by the antigen specific B cells, because the frequencies are a little bit too high. So again, this may be something that has to do with chronic responses. Another peculiar thing that we can see, or interesting thing, is that we have lower somatic type mutations. So even though AMPAs have high somatic type mutations, when we look in the global repertoire, we see lower somatic type mutations in RA patients, and we can see that in the IgG positive cells, both in the lung and in the blood, and this has also been shown by other groups. And one thing that this may point towards is again chronic responses. You may have class switching without affinity maturation, increase extrafollicular responses, and there's been a suggestion that this is connected to double negative B cells. And in some exploratory CyTOF studies looking at many markers of phenotypic markers of B cells in RA patients, we do see that there are changes in the naive B cell compartment where we have an increase in certain naive B cells. We have a decrease in class switched kind of standard IgGs positive cells, and we have an increase in double negative B cells. So there are some kind of phenotypic global shifts going on in RA patients as well. Right, so just to summarize, by using monoclonal antibodies, we have gained much more understanding in the clonality and the autoreactivity of these anti-modified protein antibodies, and we can understand that the carbamylation acetylation reactivities are really flavors within the citrulline reactivities. There are also different flavors of MDA reactivity that I didn't have time to go into here. And of course, it is very interesting to think about the B cell or the autoantibody responses, how they evolve during the risk phase, where you may have a constant autoantigen stimulation, an increase in the multi-reactivity, increase in the Fab glycosylation. Maybe there is also a shift in the balance between pathogenic and protective clones and the dysregulation. And maybe this process is more dynamic. So maybe we should think more of a balance between different clones. There's a lot of work to be done to understand the different autoantibody profiles that we see in our patients, how they may relate to the disease in the patient's disease progression, and also treatment responses. So still some work to be done. So of course, this is efforts by a lot of different people that we're collaborating with, and a lot of different groups at Karolinska and also externally. And of course, special thanks to all my group members that have generated this data. So thank you very much.
Thank you very much, Caroline, for a very fascinating talk. We have at this point one question that I will read to you from Susan Fink. Thank you for sharing this interesting data. Is the anti-inflammatory versus pathogenic effect of those monoclonal antibodies related to Fc glycosylation and/or subclass?
Yeah, no. The antibodies that we have produced, they all produced with the same constant regions, and of course, when we produce them recombinantly in the hex cells, they may not have exactly, perfect glycosylation as what we see in the serum. But we have done mass spec on these antibodies to make sure that there were no differences in the Fc glycosylation. There were no significant differences between the pathogenic and the protective. So otherwise, that would be an interesting explanation. And yeah. They're the same constant region, so they're the same isotype.
Yeah. So another question from Mark Wenner. Thank you for these fascinating studies. The effect of ACPA to prevent arthritis is unexpected. Have you others given ACPA with rheumatoid factors?
Yeah, no, we haven't done that, but that is definitely on our list to do. I think that will be very interesting to see what happens if you, and particularly then maybe IgM rheumatoid factor that will help form immune complexes. What will happen? Will they still be protective, or so we'll see. So that's something that we're gearing up to do.
I'm going to follow that up with a question of my own. I remember a number of years ago, there was quite a bit of discussion about physiological citrullination of the FC portion of antibodies, and whether perhaps rheumatoid factor would have citrulline specificity in binding to that region when it's citrulinated. Do you think that's still valid thought?
I think, I mean, we have shown that some of these AMPAs can bind to modify the FCs of other antibodies, but I don't think that that's you know, a very probable explanation to explain some of the pathogenesis. But I think it's not impossible that there will be these antibodies that are the AMPAs will also bind if you have citrulinated FCs physiologically, because there are AMPAs binding to pretty much any citrulinated epitopes. You just have to find the right clone and they will have a specificity. So that's something that I think exists, but if it's important, I don't know.
So here's a question from Jane Buckner. Do you know if there are individuals who are AMPA positive but do not progress to RA? And do they have different specificities or antibody characteristics?
Yeah, no, that is definitely something that we're currently working on. So together with also Carolyn, where we have a cohort of risk individuals. And it is hard to say if they will ever progress into RA. There are definitely in some cohorts patients that will seroconvert and go back to being negative or having low levels. There are definitely individuals that are during the follow-up so far, 2 years or so, they have not progressed, and longer time than that. If they will ever progress in 5 years or 10 years, very hard to say. But we are definitely looking into if we can see any specific reactivity patterns in these individuals that are not progressing compared to the ones that are progressing kind of earlier, at least. But we have a difficult question, but yeah.
We have an additional question here from Mark Wenner. The increase in unmutated B cells in RA patients is also unexpected to me. Is this present at baseline versus effect of immunosuppression and treatment?
Yeah, I don't think that it is an effect of immunosuppressive treatment. We couldn't see a direct correlation with that, but we have not had opportunity to look in B cell repertoires of untreated patients. We did see it though in the lungs of the early RA patients, and they are not treated. So I would then go back and saying that I don't think it's related to treatment. I do think it's something that is happening in the B cell repertoire. If it is, something that is there at disease onset or earlier, that is difficult to say, but I don't think it's related to treatment, at least.
So I have another question regarding the epitopes. When you make synthetic peptides, they are floppy and can adopt any swing of the side chains. Do you know if in intact citrulinated proteins, if the AMPA would recognize them with the same broad specificity, or this harkens back to a discussion a few years ago about whether epitopes in proteins that are citrulinated in rheumatoid arthritis tend to be disordered regions as well. Do you have any, what's your current thinking of that?
Yeah, so no, I think that's true. I think the peptides that are using are surrogate markers. They're not kind of the real antigens. And many of these peptides, some of them are designed to be in unstructured kind of loops and so on in the kind of original protein that they come from. So they are then likely to be actual targets, while other peptides may not at all have the same structure when they're in their protein backbone. And that's why I also think it's really dangerous to, when using these peptides, thinking that they are reflecting a response to fibrinogen or vimentin, because that's not necessarily true, because the epitopes may look different. But we do have multi-reactivity also looking at full-length proteins. We have done some experiments with that, with the standard different antigens, different histones, different H&R&Ps, and vimentin and fibrinogen. And you'd see a multi-reactivity, and you see different patterns in different clones. But that, not sure that is 100% related to the peptides. And I think that there will be clones that definitely have a preferential for these unorganized, disorganized epitopes in the proteins. And again, we do have a lot of antibodies that are recognizing glycine-containing motifs, and glycines, they are disrupting structures, or they are in flexible regions. And there are also lots of peptides that are containing serine, and which also will be more flexible regions. So there's definitely something there, and maybe those are also epitopes that are preferentially citronated by the pads. That's also something to think about, which epitopes are actually citronated in reality.
There are two questions from Gordon Starkebaum. The first one is, in your controls, there were several with high ACPA. What happens to them?
In my controls, in population controls.
So healthy controls.
Yeah. So I don't have follow-up on these. So these are population controls. So they are really matched individuals, in to the RA patients. They're actually from the same region and born the same year as some of the RA patients, and they are just regular people, more or less. And some of them will have some citrulline reactivity, in particular to individual peptides. We don't have a follow-up on those. They fill out questionnaires saying that they have no rheumatologic disease and so on. But if they will develop something, or if they have some other condition, we don't really know. But there are, CCP kits, there will still be 1% positivity in the general populations. That's how these things are designed.
So the second question from Gordon is, do the B-cell immunosuppressive clones work in other models besides Kaya?
Yeah, there's also something that we are working on, and maybe not. There will definitely be differences, I think, and we are trying to understand what is the critical kind of inflammatory process that are inhibited in the Kaya model, and we're in parallel working with other arthritis models and also other models of inflammation to see what's happening.
Is anybody working to take such antibodies into patients?
So there is a Dutch company that working with therapeutic AMPAs. They have developed, so they also did a long time ago, it turns out, some initial studies looking at AMPAs from phage display selections, and now they have developed some citrulline reactive clones from urine immunizations that they are driving as a therapeutic agent. So yeah, maybe.
We'll see one day.
Yeah.
We have a question from actually two questions from anonymous attendees. The first one is, there's evidence that IgA ACPA prevalence increases in normal population with age and more in women than men. Mm-hmm. Does your data confirm, and what do you think is the mechanism?
Mm-hmm. That's interesting. I don't have any data supporting that. We haven't looked at correlation with age or in the general population at IgA positivity like that. But I mean, that's super interesting. If that, you can speculate that there are some functions that will generate these kind of responses, and they may not be pathogenic but I don't know, or it's just a consequence of age and encountering a lot of citronellation. But that sounds super interesting. I want to look into that.
So the final question here is, do other autoimmune diseases also have these multi-reactive monoclonal antibodies, and are they AMPA reactivities?
No, I don't. I think this is quite unique for RA. I haven't heard that there are other of these kind of multi-reactivities. There are autoimmune diseases that have autoreactivity towards post-translation modifications. And then, if you have an autoreactivity towards only the post-translation modification, that may occur in different contexts. But I haven't heard about any other example that looks like RA. And it's not, regular immune responses do not look like this. There are some immune responses to hapten or to carbohydrates and so on, but they are generally not definitively matured and with high somatic height mutations. So in my opinion, I think that the RA situation is quite unique and interesting.
Yeah. Okay, we are a little bit over time, so I will thank you one more time for really interesting seminar, and looking forward to a discussion with you in a moment. So thank you.
Great. Thank you so much. It was fun. Thank you.